For those of you who are wondering, I'm not Christian Grefkes, because he unfortunately couldn't make it to Glasgow on short notice. He sends his regrets and wishes the presenters and you a very fruitful symposium. And that also gives me the task to say a few lead-in words. What is this DGKN OHBM Symposium? The DGKN is a Deutsche Gesellschaft für klinische Neurophysiologie, German Society for Clinical Neurophysiology, that now has somewhat of a tradition in a partnership with the OHBM. And this symposium is basically two symposia, because we had a very similar one with most of the speakers being the same actually, this year in Würzburg, well, mainly virtual to be honest, because that was in spring, and in spring things were even worse than they are now. And it was a very nice success, and now we have the second coming of the Symposium on Precision Medicine, and I'm very glad to introduce you to the podium, Thomas Geo from Singapore, Susanne Weiss from Jülich, and Gail Varaco from INRIA, and enjoy. Thank you.
Hi, everyone. So I'm super happy and super excited to be here and to see you all. So I'd like to talk today about measuring mental health. And the point being, right, if I show you a brain image and I ask you simple questions, how old is this person? Is she at risk of developing Alzheimer's disease? Is she depressed? Well, it's pretty hard to answer those questions, right? We're going to need fairly sophisticated analysis to get the information from this image. And so one framework to do this is to use machine learning to predict a mental health outcome. And so first I'd like to talk about the vision and to convince you that we can extract non-trivial imaging biomarkers. Then I'd like to talk about possible shortcomings, in particular the fragility of evidence for prediction. And then I'll give broader vision on validity. And so first, let me try to convince you that we can capture very non-trivial phenotypes of mental health in brain images. And I'll take the example of autism spectrum disorder, and it's just one example. And here the challenge is that we're looking at a spectrum disorder where the diagnosis is based on symptoms. It's not based on biology. And so to assess a bit, how strong a prediction could be, we ran a competition a few years ago. So we gathered fairly rich data, so both cortical thickness and resting state fMRI on 2,000 individuals, and we opened up a competition with some pretty nice prizes for people who could do the best prediction of autism's status. And that stayed open for 3 months, and we had a few dozen participants, a few dozen people, challengers, participate to the competition. And at the end of the day, what we get is a trustworthy assessment of what people can do. And the best performers got something like 0.8 area under the curve prediction, which is a very useful prediction, at least a very useful prediction score. If we could actually, predict mental disorders with this kind of accuracy, it'd be impressive. So we did a bit of post-hoc analysis to learn as much as we could from this prediction. And maybe one of the things that really stuck out was the fact that maybe the most important factor in terms of how well those models would predict was how much data we could feed them in. And we're looking at 2,000 individuals, and here I've shown you the curve, feeding more data and extrapolating it. And the kind of extrapolation we get is that if we had 10,000 individuals, we'd get an AUC of almost 0.9. Now that's an extrapolation, it may or may not be true. But my take-home message here is that the amount of data for this task is currently the limiting factor. And interestingly, not clearly the disease heterogeneity. Now we looked at what was important in the data to predict, and the way we did this was that we removed different modalities. And interestingly, what stuck out on this specific problem was that functional, so resting state fMRI, was significantly more important than anatomy. Now, I don't claim this as a general finding, but it seems that to predict ASD status, it's the case. And this was true across multiple prediction pipelines. So I hope I've convinced you that, given enough labels and data, machine learning can, in certain cases, extract non-trivial biomarkers of mental health. Now, can we trust all the biomarkers that are predicted? And I'll show you it's maybe a bit more complicated. And the first thing is that assessing predictions absolutely requires unseen data. Here I'm showing you different models. The red one being the more complicated model. It fits the data perfectly, but quite clearly, it's absolutely unable to extrapolate to new data. So the only way we can judge a model is by bringing new data, and not by assessing data fit on the data we already had. So the way this is done in machine learning is fairly standard. We typically, we split the data between a train set and a test set, and we might do this many, many times, and we might call this cross-validation. And the people who participated in our challenge did exactly this, to choose their model. And so we had given them only part of the data. We had given them only what we call the public set, and we had kept some data private. And so what you can see on this graph is that some people were incredibly optimistic on the public set. They had amazing predictions on the public set, but really bad prediction on the private set. And now this is a bit worrying because these people were specifically not incentivized to do this because they knew that they would be judged on the private set. And if we look at how it happened during the competition, at the beginning of the competition, people had models that performed as well on the public and the private set. And as they kept working on those models, some people improved massively the prediction, the quote-unquote prediction, on the data they had, but not at all on the private set. So these people were running cross-validation and were quote-unquote improving their cross-validation results. But what they were really doing is overfitting those cross-validation results. And here the problem is that cross-validation is imperfect. It's noisy. And this can be easily understood. And I've been rambling about this for quite a few years. And the point is simply sampling noise. If I have 100 labels in my test set, there's only so much information you can get out of these 100 labels. And if I have 200 labels, well, I have more narrow confidence interval. And 1,000, I have more narrow confidence interval. But those confidence intervals are still, around plus and minus 3%, and that's 95% confidence interval. So now if I start, going in the tails, I can go much, much further. And people do this, as we've seen in the competition. And here, the problem is that I can vary trivially things in my prediction pipeline, and this is what I did here a few years ago. I just trivially varied the amount of smoothing, arbitrary choice of predictors, and I observed massive variations on the observed cross-validation score. Even though on this specific data set that I created, there's no information. The ground truth score is 50%. You can't predict. So there's a danger is that as people try things out, they're just exploring sampling noise. And then they're going to pick the best one. But the best one is just chance. Now, if you look at what happens in the publications, what we can see is that the larger the study, the smaller the reported prediction accuracy. Now, there might be cell explanations to this, but I'm worried that when people have better cross-validation simply because they have more data, they're less optimistic. They're less falsely positive. And so that basically the effect that we've seen in our challenge is just rampant in the literature. And add to this the fact that the incentives are not very well aligned in the literature. You're more likely to publish if you report a big number. So, this has gotten me a losing sleep for quite a few years. Okay, so the problem here is that small sample sizes give us wiggle room that completely kill my trust in publications. And that's a fact. And I review papers, and those papers have smallish sample sizes, and I just don't want to be reviewing those papers because I'm tired of being unpleasant to the authors, to be honest. Okay, so let's try to think broader, and here the vision I'd like to push is that if we think about bigger data sets and about clinically useful settings, we need to rethink how we do our studies. And I've stressed that we really need a lot of outcomes. We really need a lot of labels. But most individuals do not have a diagnosed condition, and that's a good thing. So for instance, if we take UK Biobank, which has 500,000 people, and if we look at the number of people in UK Biobank with Alzheimer's disease, it's approximately 500. Schizophrenia, approximately 500. So even on that huge, huge data set, we simply do not have many diagnosed conditions. So we need to rethink how we work, and we need to turn to new outcomes. One example of a useful outcome is that of brain age. And this is something that's been going on for quite a few years, and I think there's a parallel symposium on this. And the idea is that you train your predictor with chronological age, but what you're really trying to target is aging. And there's a difference between age, chronological age, and some form of biological or brain aging. And what the predictor gives you is the expected age given the image of the brain. And then you can look at the discrepancy between this expected age and the date of birth of the person, and some people call this the brain age delta. And we and others have shown that it captures interesting information, such as correlating with cognitive impairment. So, this is a biomarker that's been learned from a surrogate outcome. It's not directly clinically a relevant outcome, but it still captures useful information. And we tried to push this agenda further and to push forward the idea of proxy mental health measures. And the idea is to use machine learning on imperfect correlates to build biomarkers of mental health. And here I use the definition of biomarkers that's given by the NIH, for instance, which is objectively measured characteristics. Now, we could debate about objective, but that's not the point. And the point here is that we can extract these despite the lack of reliable diagnostic information. And we can even do this as a multimodal setting that captures information about the patient on very different sources of data. And we can apply these to a wide population, not in a specific disorder. And then we can use them to solve all kinds of problems such as treatment development, public health policy, or personalized medicine. And so we did this for aging, and I already discussed this, and we tried this for neuroticism, for intelligence, and so we applied machine learning to brain imaging and to sociodemographics, and the important thing here is that when we're training these things to reconstruct neuroticism or fluid intelligence, just like age, the measure is an imperfect one. The measure of neuroticism, the measure of fluid intelligence is a measure that's done with a questionnaire. It's not a gold standard, it's a silver standard. And so what we get is those proxy measures, those approximated measures, and they correlate, they associate very well with real-life health behaviors, such as the amount of alcohol you consume, the amount of exercise you do, the sleep. And the interesting thing is that it actually associates better than the original questionnaires that were used to train, or the original age measure. So, there's this notion that the proxy measure is capturing more than the original measure, just like with brain age. And one important thing here I find is that what we're doing needs to have some form of external or ecological validity. You know, we can't validate those measures with the same questionnaire that we use to train them. That's totally circular. We need to show that they're useful for a broader question. And so now we can look at the importance of brain imaging in this endeavor. And here I've shown with a dotted line the prediction without the brain imaging, and with a full line the prediction with the brain imaging. And bigger is better, so you can see here that brain imaging gives us a better estimate of people's age. And not only, you know, their income. Income is a good predictor of age, by the way. And so we can look at, you know, the other settings. And this is a bit of a depressing story. But basically, brain imaging does not help giving a good assessment of fluid intelligence. It's more like the years of education that give the best assessment of fluid intelligence. Basically questionnaires that on people's sociodemographics. Same thing for neuroticism. And what I learned here is that we really need to give more importance to the sociodemographic data when we do image analysis. Okay, and this is kind of the end of the story that I wanted to make. So machine learning can work. We do need to be creative about those labels, but too often we're doing this on too small sample sizes. And also too often, we're focusing on imaging and forgetting all the rest, which is absolutely crucial. And this is a wider discussion, and I have a poster about this that I'll be presenting on Wednesday that is entitled How I Failed. Thank you.
I think we can have time for a quick question.
Okay, so there are a bunch of questions. One of them was the anatomy performed badly, but presumably this was all T1 weighted data with a terrible contrast for subcortical nuclei. Rather than collecting more data, maybe we should collect better data. So I get this question all the time. And that's a question of internal validity versus external validity. We can strive for better data. We can strive for the perfect experiment, the 15 Tesla scanner. And then we're going to be scanning 3 people, and I'm not sure we're going to do something that can go outside of the lab. The older I get, the more I'm interested in getting outside of the lab. So it's a philosophical question. There are many, many, many questions. So why do these competition always gives money to the best prediction and not to the one which allows new explanation? What's an explanation? How do you judge an explanation? Well, the way we typically judge an explanation is, well, it matches what we already know. I don't need a competition to match what I know. Is brain age simply a difference detector with nothing to do with biological aging? The more different your brain, the older your brain age delta? That's an interesting question. It's a one-dimensional analysis, so you're going to be capturing. Sometimes you're simply going to have outliers, and they won't mean anything. So clearly, you can't reduce an individual to his or her brain age. Whether this gives you an interesting information is just like temperature. You know, we measure temperature at an individual level, but it captures all kinds of different things. I still hope it can be an interesting objective measure, and people have shown very beautiful thing. Sylvia Villeneuve has a very beautiful study where she links brain aging to Alzheimer's disease in a very convincing way. Of course, you know, it's an open debate. I'll look at only one question and then leave the stage. And I need to understand the question too.
Kyle, maybe we can move on to the next speaker and you can answer later. Yeah.
Very good.
Okay, so hi everybody. So my name is Susanne Weiss from Research Center Jülich, and I'm gonna basically follow up on Gail's talk, kind of readdressing some of the points from a slightly different angle, but also basically with the main point being that yes, we can be really excited about all the possibilities that we have with predicting mental traits from neuroimaging data, but there's really, really many points that we need to be aware and be careful about. Okay, first of all, why would we even be interested in predicting any mental traits? Well, quite straightforwardly, we're all different, especially with respect to mental attributes, personality, clinical factors. And we cannot just look into another person's head and interfere these mental attributes. We can try to approximate them by doing interviews or by doing some kind of tests, but we cannot really get an objective measure of another people's mental traits or mental attributes. And there's the hope that with this neuroimaging data, that we're collecting lots and lots of neuroimaging data of bit better and better quality, that might actually maybe bring us a bit more to an objective estimate of the mental traits rather than the classical interviews, behavioral testing. Obviously, it's not a new idea to look at individual differences. We have seen lots and lots and thousands of graphs like that, so this is basically only a constructed one, which would show that in some certain tasks, women perform significantly better than men on some random task. And we would see a highly significant result here, a very big difference, but would that be any use as a biomarker, or would that be any use to predict anything about an individual? And looking at the same constructed data in a more honest plot, we see, no, it's not actually useful, because there's many men that perform really higher than many women. There's many women that perform really low. So actually this statistical difference between these two groups cannot tell us anything about individuals, so it's not really helpful for any biomarkers. On the other hand, another constructed example, we could have a variable, for example, the height comparison of basketball players and sumo wrestlers, and we do not see any difference there. There's no significant difference, nowhere near significant. We can also look at their weight. Again, we do not see any difference there. But if we put these two variables together, we see a really clear separation between the two groups. So these multidimensional patterns can really greatly increase predictive power over just looking at one variable. But as already mentioned, it's really, really prone to overfitting, and as Gail already mentioned, the out-of-sample validation is a critical thing. And especially that's the case when predicting biomarkers, especially in the clinical case. We want to know something about individual cases, and we want to know something about individual cases outside of our training group. So the basic approach that we use is we have a training group, we have some features, for example, neuroimaging features, could also be behavioral features, and we have some target variable. We put these into a model, we learn these, tell the model learns to predict the target from the features in the training data, but the really critical thing is then to also have an independent, completely independent test group to validate the prediction of the target if that also works in the independent group. And luckily, because not all of us are all into this, and there's a lot of steps like controlling data leakage to cross-validation or optimizing your parameter, but now we have a lot of toolboxes that are coming out that at least help with these critical steps. Obviously, you still need to know what you're doing, but they can be helpful to address some of these caveats of the machine learning. Okay, as a first example, again, brain aging. So this is an example where 128 different workflows were used. So there's different ways to extract features from a structural brain image. There's different models that you can use, and testing a large number, so 128 different workflows, it was possible to predict the age with a really good accuracy, both with inside and across site. So there were, as Gail pointed out, we need lots of data. There were different sites, different big data sets, but both within and across site, the prediction worked really well, which is nice to see. And that also has clinical relevance, keeping in mind all the critical points that were made in the first talk, that we can see that for an Alzheimer's disease patient shown in red, the age gap, so the difference between the predicted age and the actual age, is significantly larger than for the controls shown in green, and in between we have early and late mild cognitive impairment. So indeed, it seems that machine learning can find generalizable MRI phenotype relationships, but we have this big problem with dimensionality. So the thing is, a brain has a lot of voxels, and the algorithm doesn't see a brain. The algorithm only sees several 100,000 numbers, and the algorithm doesn't know anything. The machine learning doesn't know anything about the shape of the brain or anything. It's really just a set of numbers. And even we have, with the large cohort data that we have now available, we're still likely to have, in brain imaging prediction, likely to have many, many more features than subject, and that will cause overfitting. So will cause will result likely in a model that fits on our training sample, but that will not generalize well. But of course, we are lucky because we are still a bit more clever than the machine, because we know that's not really 400,000 different pieces of information. A brain is organized in networks and in areas that activate or connect together. So that is one way to reduce this dimensionality. And that's an example where we did this. In this case, we were classifying sex of participants, and we used a brain parcellation that was established based on the connectivity during resting state of these brain regions, basically reducing the several thousand voxels to roughly 400 parcels, and then extracting the connectivity, the brain connectivity during resting state. But we still reduced it even further by not training the model on the full whole brain connectivity, but rather only training separate models for each node. So only for each node we had 435 features then, and for each node or for each parcel we did independent models, and that also has an advantage that we can see which parts of the brain distinguish particularly well between the sexes, and we can relate that back to certain cognitive domains. And this approach is not only possible for binary classification, it can also be applied to continuous variables, like, for example, here, executive function predicted based on resting state brain connectivity. So yes, individual prediction in new subject is possible. So this is actually shown on the test data set, which was completely independent from the training data set. And we can do this for a variety of different phenotypes, binary ones like sex, age, cognitive performance, several ones. So very nice, very promising. But another thing we need to be aware of is this batch effect. Again, we're dealing with big data sets, but big data sets acquired at different sites. And we see quite a nice cross-validation. So this, again, is a sex classification. And that worked really well in the cross-validation within the data set, but it didn't work particularly well at all when we applied this model to data that were acquired at different sites. Because not only the model needs to be generalizable, but here we additionally have the problem that we also need to control for non-biological variability in this batch effect, simply because the data are acquired on different scanners, causing a lot of variability that we do not actually want in our models. Another way of dealing with the problem of dimensionality is using meta-analytical networks. So instead of again, looking at separate voxels, we extract based on the literature. We extract using statistically derived meta-analysis. We extract networks and network peaks, for example, for working memory or emotion, language, whichever, and then look at the connectivity within these cognitive networks, these meta-analytical networks, which are much smaller in dimensionality again. And that also has the additional advantage that if we have resting state data, which can be really easily acquired, relatively straightforwardly acquired in patients, for example, we can look at different meta-analytical networks within the same data and see which are best suited for the particular prediction we want to do. And this is just one study where we looked at the size of the networks. So we have the network size on the x-axis. So the bigger the networks, the identification, so this is not prediction, this is how well can I identify one subject based on the activity or connectivity within these networks. And the larger the network overall, the better this identification works. But as opposed to random networks shown in blue, our meta-analytical networks always perform better. So it makes sense, there is some additional information in these cognitive networks rather than in any just random networks. And also, this is just an example, we can also not only use resting state data, but movie data, naturalistic viewing, where people see a movie instead of just lying in the scanner left by themselves. And we see that, for example, for the social affective networks, that is different between these movies. And if we were to use another meta-analytical network, we would see another pattern. Again, we can use this to optimize our predictions. So nice, different features, different meta-analytical networks, for example, from one single neuroimaging data set, makes it much easier to apply this in the clinical context. Okay, more caveats. Not only do we really look into the features that we're having, we also really need to understand the sample that we're looking at. So this is an example predicting hand grip strengths based on brain imaging data. And this looks really, really quite promising, quite a big R, looks nice, looks like the prediction works. But if we look at the data in a bit more detail and actually separate it into males and females, then we see that within the sexes, predictions do not work quite as well at all, and rather the overall prediction is driven by the sex difference rather than the actual individual brain activity. So quality of models depends on the targets, the features, and also on our sample characteristics. Very important to be careful about that. Still another caveat, bias. It's really easy to get biased models. And this is a very basic example where it's quite obvious, really. We were, again, predicting, or rather classifying sex based on structural imaging data. And that worked. We used a matched sample matched for age, and we extracted gray matter features and predicted and found a classification accuracy of 97%. Great. Looks great. But then we looked at the data in a bit more detail. So these are male brains, red are female brains. This is the label that algorithm assigned to them, male or female, and this is brain size. And it's really, really obvious in this case that what the algorithm did, the bigger brains were classified as male within both sexes, and the smaller brains were classified as female. So nothing about individual differences in the brain, but only brain size drove this prediction. So really biased model, which is obvious when you go back to look at the data, the training data. Female brains are simply much smaller than male brains, but it's important to keep in mind. And it's one relatively easy way to correct this problem. There's others, to use a training sample that is not only matched for age, but also for brain size. So we selected males and females with matching brain sizes, and then we see, okay, the model does not show this bias anymore. The prediction is not mostly based on the brain size. And it still has quite a good accuracy of 87%. Okay, keep in mind models reflect biases and limitations in the training data, so be very careful about the data. The final point that I want to make, how do we interpret these findings? So this is an example where we predicted openness from 3 different naturalistic viewing conditions from the HCP data set and from different cognitive tasks, and it seems in this case, okay, this particular movie 3 predicts very well, the language predicts very well, and then we can technically easily extract the features, so the regions of the brain that these predictions are based on, which are the most important features to achieve these high prediction results. And that is nice, but it's still very technical, so that still does not tell us anything about the individual within these groups. And it's also still, at least if you go to the clinical case where we're dealing with patients, and probably people not dealing with machine learning at all, are these explanations, are these technical identifying best features, is that actually enough to convince any non-experts to trust in what we're doing there? So model interpretability is relatively easy, just extract most important features, but results interpretability, especially for the non-expert, is a completely different thing. Okay, and that's the summary, but I think I have shown you. So machine learning is very promising. We can find generalizable relationships. We can predict from neuroimaging data across a variety of phenotypes. And we can, if we do it in a clever way, we can use this based on basically only just a single resting state or maybe naturalistic viewing scan. But we need to be aware of batch effects, differences between the scanning sites. We need to watch very closely our targets, our feature spaces, and the samples. We might have biases in our data, in our models. We need to be aware and check for that. And we need to really work a bit more on the interpretability of the results. Okay, and that's it. Thank you much for your attention, and maybe there are questions.
So I think we have time for just one quick question. Actually, there are many good questions in the app, but I think some are suitable for multiple speakers. So let's just go with something that's specific to your session. One question that they have, because you mentioned about multi-site, the question is, what is your view on multi-site harmonization to remove batch effects, for example, using combat?
Yeah. I mean, certainly that is what most people are doing, and that's certainly one way of dealing with it, certainly. But I'm still, personally, I think there might also be other ways to actually include these batch effects more in the actual model creation themselves, and we might want to look more into this instead of doing basically the harmonization on top of it.
Okay, thank you. So let's thank Suzanne again. Yeah, perhaps adding on to what Suzanne's answer to the question, I think Gaia has shown that, for example, if you train from multi-site data, then it would naturally generalize well to new sites, even without harmonization. So it's possible that if our goal is to predict out-of-sample data, then we might not need harmonization if we are training with multi-site data. Yeah. So for the third speaker, unfortunately.
So unfortunately, thank you. Yeah, unfortunately, the third speaker couldn't join us, but she has a video for us. So we're now going to hear Geza Hartwigsen. I hope I'm pronouncing this somewhat correctly. Can you play the video? Thank you.
Dear Christian, thanks for organizing this symposium. So in my talk, I will discuss the role of domain general networks for language reorganization. And I will provide examples from the combination of plasticity-inducing neurostimulation protocols and neuroimaging to investigate short-term reorganization within and across networks, mainly in the healthy language network. What do we know about short-term reorganization? I have recently suggested a model for flexible redistribution in the language network. And one core assumption of this model is that after large-scale disruption of the specialized network induced by neurostimulation or by a brain lesion, we may observe compensation via recruitment of domain general networks. So such networks may support the maintenance or recovery of function. How should that work? Let's assume we have a disruption of a key region for language induced by inhibitory neurostimulation or a brain lesion that results in a decrease of the contribution of the specialized network. Now, in some cases, we may observe a stronger recruitment and upregulation of domain general networks that helps to maintain or regain function. Domain general functions such as attention, cognitive control, or working memory are known to support all higher level cognitive processes. And the key idea here is that if the specialized network is decreased in its contribution, stronger recruitment of such domain general networks may to some degree compensate for the disruption without, of course, completely replacing it. Now, one question that I'm currently interested in is whether such short-term reorganization via plasticity across networks is a key mechanism of reorganization across different brain states, that is in young and aging and in diseased brains. And I think a better understanding of such flexible redistribution may help to develop more effective treatment options with neurostimulation protocols. So what do we know in the healthy young brain? I will first discuss targeting language-related brain regions to map plasticity across networks based on recent data from the reading network. So a first study led by Sabrina Turka from my lab Here we were interested in short-term plasticity during reading in the healthy brain. We know that reading engages a distributed network of mainly left hemispheric areas. We reasoned that if we target the left posterior superior temporal gyrus, so a key reading area, then we may decrease its contribution, which may result in compensatory short-term reorganization. So we had healthy participants perform reading tasks of pseudowords and words after receiving TMS. We observed that inhibitory RTMS over the left PSDG slightly delays pseudoword reading. Now the question is, what are the neural correlates of such modulatory inhibitory TMS effects? We observed strong inter-individual variability at the neural level and no clear pattern at the group level. So there was no evidence for changes in task-related activity at the stimulated area. And what you can see here is the RTMS effects on task-related activity during pseudoword reading plotted as overlap for at least 2 subjects. You can see that there was strong variability here. Some subjects showed an upregulation of areas mainly outside the reading network without any clear pattern or a strong overlap between many subjects, whereas other subjects showed a decrease in task-related activity or downregulation, but again, no clear pattern. So in the next step, we used the multi-voxel pattern analysis because such multivariate approaches should be more sensitive to capture the modulatory TMS effects. That is something that we have shown with different language tasks before. And indeed, the MVPA showed a shift of the task-related activity during pseudoword processing. So inhibitory RTMS over left PSDG, so in this area, shifts task-related activity towards IFG and IFS, so an area associated with phonological or general working memory processes, likely a domain general contribution here. And finally, we were also interested in changes in the effective connectivity and the modulatory influence induced by TMS. So we performed a dynamic causal modeling analysis in a predefined reading network, and we found that RTMS over the PSDG increased the facilitatory drive of the left ventral occipitotemporal cortex on the targeted PSDG. And we reasoned that this increase in the facilitatory drive after disruption of the other node likely reflects compensatory reorganization, which would be in line with previous studies from our group and others showing an increase in the facilitatory influence of another area on the targeted area after disruption that was behaviorally relevant. So in this case, the behavioral relevance remains to be shown, but we think that these results together provide some first evidence that both language-specific and domain-general short-term plasticity, so domain-general working memory areas and changes in the connectivity inside the specialized reading network can be found in response to inhibitory neurostimulation of the reading network and may help to maintain task function, may be compensatory here. Now, in the second study that is still ongoing, Sabrina is interested in short-term plasticity during reading in people with dyslexia. Dyslexia is characterized by reading impairments and often associated with an underactivation of the specialized reading areas. So we reasoned that if we apply facilitatory TMS over the PSDG, a reading area, then this may reshift the language-related activity and should be associated with better behavioral performance in this people with dyslexia. At the behavioral level, we found that facilitatory RTMS of left PSDG improves the overall reading accuracy in people with dyslexia in the task that was performed in the scanner after TMS. So they became better in overall reading of words and pseudowords. Now, what are the neural correlates of such improvements? We found that at the univariate level with univariate analysis, RTMS decreases task-related activity in right hemispheric domain general areas during word reading. So no effects in the specialized reading network, but a decrease of the task-related activity in the right IPS and SPL domain general area. And such decreases in task-related activity likely reflect decreased task demands. This is something that we have observed during other language tasks with anodal stimulation during fMRI. If the task load is decreased due to facilitatory stimulation, we see less activity in these multiple demand areas. And here MVPA results could show specific changes induced by neurostimulation that were task-specific, so specific for word or pseudo-word reading. For word reading, we observed shifts in task-related activity in right precuneus and IPS, so this area that was very close to the one observed during univariate analysis. For pseudo-word reading, we observed shifts in task-related activity in the right insula and central operculum. So it seems that RTMS of left PSDG modulates task-specific activity outside the specialized reading network in domain general areas associated with cognitive control and general processing demand. And such changes are highly task-specific, are different for words and pseudo-words. So overall, facilitatory RTMS effects in people with dyslexia are underpinned by task-specific changes, likely reflecting decreases in domain general areas, which points towards the role of such domain general areas under increased task demands in people with dyslexia. And maybe this also suggests that targeting such areas with facilitatory neurostimulation could be used to aid treatment or training protocols in such impairments. Now, in the second part, I want to move to targeting domain general brain areas to map plasticity across networks. So this is data from an fMRI study led by Ole Numsen from our lab, where we investigated domain general network interactions across different cognitive domains. So we focused on the inferior parietal lobe here, an area that is part of the default mode network, so this task negative network, but this area is also associated with different key domains of human cognition, language, attention, and social cognition. And based on tasks from these three domains, we were able to distinguish two regions in both hemispheres, an anterior one and a posterior one. And now we found that the functional connectivity profiles in these subregions differed and were highly domain specific. So we had differences in the connectivity profiles for attention, semantics, and social cognition with respect to its connectivity with the major brain networks in the rest of the whole brain, some of which are also described at rest. And this shows that even though we have a domain general area here, its connectivity profiles are modulated based on the specific task requirements. And now the next step is to target these areas with neurostimulation and see if the changes in the modulatory effects are also task specific to better understand the task general versus specific contribution of these subregions. So this is work again led by Ole Numsen and Katie Williams from my lab, where they investigated task-specific effects of network stimulation in the default mode network. And this is work in progress, but some preliminary results suggest that the induced electrical field strength in the IPL is strongly correlated with the behavioral modulation induced by TMS in a highly task-specific manner. So here you can see the behavioral neurostimulation effect in the semantic task that we used, that it strongly depends on the induced electrical field strength in the left posterior IPL subregion. So there was a correlation between the induced electrical field strength. The stronger the induced electrical field, the faster the TMS-induced modulation of the semantic response speed. So the stronger the facilitatory TMS effect. These effects were highly task-specific, so selectively for word relative to pseudo-word processing, and they were strongly domain-specific, so only observed in the semantic task relative to attention and social cognition, and they were also specific for the subregion. And on the long run, this may have potential therapeutic relevance because we could optimize the neurostimulation protocols, so the targeting and the dosing of a specific protocol for the respective subregion and for the process of interest that may be impaired in a certain disease. So finally, I want to talk about domain general network interactions in the aging language network, because we know that many of our patients are of older age, and aging is associated with general cognitive decline and large-scale network reorganization, and it is debated whether such reorganization is compensatory or reflects dedifferentiation. So work led by Sandra Martin from my group shows that the increased interaction in domain general networks in the aging brain is associated with better behavior in older adults. So using a verbal fluency task during fMRI, Sandra showed that older adults show generally increased task-related activity in domain general areas, including midline structures. Older adults also show increased task-related interactions between domain general areas and networks. So here illustrated for the right insula of the multiple demand network and default mode regions. And older subjects show increased interactions within the domain general multiple demand network to be associated with better general behavior outside the scanner in executive tasks. So this shows that some of the observed reorganization patterns appear to be beneficial, appear to be compensatory in the aging brain. Using more advanced analysis, Sandra could further show that older adults show increased facilitatory network interactions, which are associated with better verbal fluency behavior, so also seem to be relevant for language processing. So this is data from an ICA and correlational PPI analysis to investigate changes or differences in network interactions between old and young subjects. And these age-specific differences show an increased facilitatory network interactions between usually anti-correlated networks. So increased facilitatory network interactions between task-positive and task-negative networks. These facilitatory network interactions predict better verbal fluency selectively in the aging brain. So they seem to be of behavioral relevance, which again shows that some of the observed changes of the reorganization profiles are adaptive. And this suggests that we may target such domain general nodes with facilitatory neurostimulation to aid language recovery after brain lesions. So to summarize, what do we know about flexible redistribution across networks after neurostimulation? Perturbation of key language nodes results in strong remote effects, not necessarily at the stimulated area, and often associated with a shift towards domain general networks. Compensatory reorganization in domain general areas may help to maintain task processing at a high level. Facilitatory neurostimulation then may decrease domain general network contributions, likely reflecting less task effort. Domain general networks also show specialized contribution to different cognitive key domains as observed in the default mode network, and targeting domain general networks yields domain-specific modulation profiles as observed for the semantic task in our preliminary analysis with the E-field modulation. Domain general network reorganization also contributes to language performance in the aging brain, and some of these nodes may be good target candidates for facilitatory neurostimulation to aid recovery. With this, I'm at the end of my talk. Thanks for your attention. I wanted to have a job announcement here, so I will be looking for a postdoc soon in a project on network reorganization with concurrent TMS fMRI. If you're interested, please approach me. So that's it. Thanks for your attention.
And we now have Thomas Yeo, who is going to give the last talk of the session, and then we'll do a Q&A.
Hi. So unfortunately, because of the way it's set up, the third speaker is remote, so she won't really be able to answer questions. So what's going to happen is that after my talk, the three of us would answer questions together. In the interest of time, I'll try to go a bit faster. Okay. So this talk is divided into two parts, basically using machine learning to make sense of disease heterogeneity and to use machine learning to predict disease progression. For the first part, this is work done by Xiuming, Nambo, Si, and Valeria. So as we all know, psychiatric disorder is highly heterogeneous, so there is a lot of heterogeneity within a disease, but actually there's also a lot of overlaps across disorders. So this can be illustrated by this schematic shown here, where a single brain circuit might be involved in multiple cognitive processes, right? And deficits in these cognitive processes results in symptoms. So deficits in cognitive process 1 might result in symptoms related to disorder A, which deficits in cognitive process 3 might result in symptoms in disorder B, and these symptoms can also overlap across disorders. Now, we know that neurological disorders are a bit better defined than psychiatric disorders, but there's also significant heterogeneity within neurological disorders. So for example, even within late onset Alzheimer's disease, there can be a lot of heterogeneity, right? So even though Alzheimer's disease is characterized by memory loss, but there can be other deficits such as executive function or language deficits. So as a result, this schematic in general can be helpful for understanding the relationship between brain system processes, symptoms, and disorders. Now, this schematic is just a schematic, right? What my group like to do is to take this kind of schematic and try to develop a kind of a model, right? That mathematical model that describe the schematic, which we can then fit to data. And in doing so, by fitting the model to data, we hope to gain some insights from the model. So for example, my student Shuming developed a model to extract latent atrophy factors from late onset Alzheimer's disease. So in this model, a patient might exhibit one or more factors related to distinct patterns of brain atrophy, right? So for the important point here is that the factors can, multiple factors can contribute to an individual's atrophy pattern. So for example, patient 1's atrophy might be 90% factor 1 and 10% factor 3, whereas patient 2 atrophy might be 60% factor 1, 40% factor 2. The reason why this is important is because there are probably multiple contributors to brain atrophy, right? So for example, the spatial location of tau tangles, but also coexisting non-AD pathology, such as risk factors such as cardiovascular disease. So we believe that this is an important modeling assumption. So we fit this to data, 188 patients, and we found we discovered 3 factors, and we applied this to voxel-based morphometry data. So this model allows to estimate the probability that a patient is expressing a factor and the probability that a factor is associated with atrophy at a voxel. So we found 3 factors shown here. There's a medial temporal factor associated with atrophy in the hippocampus and medial temporal lobe. There's an atrophy factor largely associated with subcortical structures. And then there's a factor that's related to posterior cortical atrophy sparing the medial temporal lobe. Now, obviously, if you give a kind of a clustering algorithm to find patterns, it will find patterns, right? But what we want to show is that are these patterns, atrophy patterns related to symptoms, right? So, actually before that, I want to emphasize that the way this approach works is that each individual exhibit a unique mixture of factors. So let's go back to the relating the atrophy factors to the symptoms, right? So here we look at both memory and executive function decline. And we look at cross-sectional and longitudinal changes in amyloid positive, cognitively normal, mild cognitively impaired, and AD dementia patients, right? So by fitting kind of this linear mixed effects model, we're able to estimate this kind of trajectories of memory and executive function decline. And roughly speaking, is summarized in this schematic. Basically, the green line corresponds to the temporal factor, the blue line is the subcortical factor, and the red line is the cortical factor. Okay, so in this case, the green line is at the bottom, right? So it means that the medial temporal factor is the worst memory decline. Whereas for executive function, cortical factor has the worst executive function decline. And the reason why this is important is that there's a double dissociation between the temporal factor and the cortical factor, right? So the temporal factor has worse memory, whereas the cortical factor has worse executive function. And the reason why I feel that this is important is because these factors are not just capturing disease stages, right? Because of this double dissociation, but in fact it's explaining inter-individual differences in how the disease is expressed. We can extend this approach to function connectivity data. So then this allows us to look at autism spectrum disorder. In the interest of time, I'll just, I'll be brief here, right? We also show that again, these function connectivity factors are associated with ASD symptoms or executive dysfunction and externalizing symptoms. So far, I've shown you where we have looked at heterogeneity within a disorder. We can also look at overlap across disorder. And this is work done by Valeria, where essentially using kind of similar approaches, we can extract basically distinct patterns of functional connectivity deficits across multiple disorders. So here we found 3 factors corresponding to psychopathy, cognitive dysfunction, and impulsivity. And what was quite interesting is that across these factors, there were actually overlap across the somatosensory motor cortex. So the second part of this, I'm going to focus on using machine learning to predict disease progression. So the first part was more interested in gaining insights into disease nosology, trying to understand heterogeneity within disorder. Here we're simply interested in using, let's say, supervised learning to predict behavioral traits or disease progression. So in the case of Alzheimer's disease, right, we know that there's no effective treatment, and so the idea is that successful treatment should target at-risk individual early in the disease process. And so we consider the TAPO challenge, whereby we're given multimodal biomarkers at one or more time points, and we would like to predict cognitive scores, brain atrophy, clinical diagnosis for every month indefinitely into the future. In this case, we're using ATNI, so we'll only evaluate about 6 years in the future. Okay, and in this case, we develop a deep recurrent neural network that can handle missing data. So we did a nested tenfold cross-validation, and we try to predict future diagnosis, ventricular volume, and cognition. So in the interest of time, I'll just show you diagnosis, but the results are pretty similar. So in this plot, x-axis is years into the future we're trying to predict. Y-axis is the diagnostic accuracy. This is the constant baseline, so this is just predicting the diagnosis, whereby you assume that the diagnosis always stays the same. Okay, right? So if a participant start off as normal, then we assume that the participant is normal for the next 6 years, right? So it's a kind of a silly little baseline, but actually you can see that accuracy decreased further in the future. But what is interesting is that even 5 years in the future, you can predict 80% kind of accuracy, right? Which means that for this kind of prediction, especially predicting longitudinal data, you need to have a very careful baseline, because a lot of time, things don't change, right? So just predicting a constant baseline works pretty well. With our deep recurrent neural network, we perform better, and 5 years in the future, we still obtain about 90% accuracy. And in our paper, we show that it does have advantages over other approaches. We also submitted our results to the TAPO challenge. This is the results from the most recent leaderboard, where we're currently about 3rd out of 60 plus entries. So in this TAPO challenge, the held out data is something that we did not touch at all when we trained the data. Okay, so in all of this supervised prediction stuff, as something that Gael has also mentioned and Suzanne has also mentioned, sample size is actually very important. So in the TAPO challenge, we use about 1,600 participants, right? But for this kind of N-of-1 prediction, in a few, unfortunately, the sample size is still relatively small in many studies. This is from Russ Podrak review paper with Gael. The problem with small sample sizes is that it leads to less reproducible results. But actually, just as importantly, is that we need large sample to get good prediction performance. So in this analysis, x-axis is the number of training subjects, y-axis is the prediction performance. And we can see that across 4 different approaches, there's actually not that much difference between approaches, right? So the big effect we see is actually the number of training subjects. So if you want better prediction performance, a lot of time just having more subjects works really well. Okay. The problem is that a lot of time we don't have big data sets, especially, for example, in clinical populations, or for example, in like some Alzheimer's disease variant, which is a lot more rare. But the thing is that nowadays we have a lot of large-scale public data sets, right? For example, UK Biobank is about 40,000 participants, right? And yet, small studies are unavoidable. So then the idea here is that we're interested in adapting model trained on large-scale data sets to new phenotypes in small data. We're not interested in translating, let's say, a fluid intelligence prediction model from a UK Biobank to a small data set, right? What we want is to translate, let's say, a fluid intelligence prediction model from the UK Biobank to a new phenotype in a new data set, okay? And to do this, essentially, He Tong exploits correlations among the phenotypes. And in doing so, he developed this method called meta-matching, which get a very good results. Shown here on the right, you can see that with the traditional approach shown in red, you can see that there's a big prediction gap between using 100 training participants versus 1,000 training participants. But using He Tong's approach with meta-matching, we were able to get, with 100 subjects in blue, we were able to get a similar prediction performance as 1,000 subjects. So we're quite excited about this because, for example, would we be able to train a prediction model from acne and generalize it to an atypical AD population, for example. To summarize, I've talked about using machine learning to make sense of disease heterogeneity and also to use it to predict disease progression. Right now, we're very interested in translating models from large-scale studies to small data, and in general, all code available. I'd like to thank the funding and the support, as well as the people who actually did the real work, as well as our collaborators. I'm also hiring a postdoc in my group, and at our Center for Sleep and Cognition at National University of Singapore, we're actually hiring faculty positions at our level. Okay, thank you.
Okay, so thank you everyone who have asked questions or upvoted questions on the application. We do have some very voted question, and the most voted one is about the fact that many diagnoses in psychiatry are heterogeneous, and whether supervised machine learning can be helpful or reasonable here. Do you want to answer?
Sure. We can all answer. I think in this, hello, can you hear? Is it on?
Tap it.
Can y'all turn on? Oh, yeah, okay, all right, okay. So in the first part of my talk, I actually talk about using these unsupervised learning to find these subpopulations of within a disease. So that might potentially be helpful for finding, let's say, better treatment for a particular subgroup. But in my mind, for supervised machine learning, frankly, even though the population might be heterogeneous, I think supervised machine learning might work just fine. For example, let's say you have a large group of depression patients and which have gone through various treatments. We could directly use supervised machine learning to predict which, actually it's a bit hard. I'm just going to remove it. We can use supervised machine learning to predict which treatment might be more appropriate for different patients. So even though it's heterogeneous. Yeah, what do y'all think?
Maybe if I can add, you can have a very complex decision pattern. You could have, you could picture a 2D space where the controls sit on the middle, and then you have 4 groups of patients that are very different sitting on each side. As long as you learn a nonlinear classifier, the nonlinear classifier can, distinguish controls from people who are different from controls.
4 subgroups of depression, for example.
So heterogeneity and machine learning has no position. It's more heterogeneity and linear classifiers, because linear classifiers make the same decision everywhere in the space.
Yeah, there's also the question, how can unethical applications be prevented? For example, homophobic countries inferring sexual orientation for criminalization. And I think that's really a very, very relevant point, and not only in the sense of like intended misuse of these methods, but also in the sense that we really, really need to make sure that these biases and these aspects are not already in the data that we're working with, because especially like and it's just because here's the homophobic, the sexual orientation. there's a lot of data missing about that, and the data might have been already collected with certain bias, so that's really an important factor.
On the problem of ethics, it's a bigger one. I work a lot on machine learning in many applications, and it's been used to do bad things. I'd like to say that we have the Manhattan Project, a dilemma. We can choose to let other people build the technology, or we can build it ourselves. Somebody's going to do it. So we might as well do it as a community and set the standards with which it's done.
Yeah, I don't have much else to add, but I guess for this kind of ethical issues, what is important is that the people working on this should itself be diverse as well, because they would foresee, the problems that this technology can bring. So, so I'm hoping that with, increasing OHBM focus on increasing the diversity of our society, our organization, that we could help alleviate this problem, where the diverse voice speak up about how these issues might be problematic. Let's go to a kind of a hot question about what is not smallish n? How big of data is big data? Is big enough? Gael or Suzanne?
I would just first of all say it depends.
Yeah.
It depends very much on what you're trying to do. And yeah, but in general, bigger is better.
I guess there are two things to keep in mind. One is what's the effect size that you're trying to learn? And some people have commented that 10 subject might be enough. And that's true. Some things are glaring and you can see them easily. That's the first thing. The second thing is, how can you get external people to trust your results? And this is where it's a problem. There's massive analytical variability. There's, we can torture the data in gazillion ways and get gazillion answers. And having been in this community for quite a while, I don't care how you torture your data. If you show me that you can, on new subject, generalize something non-trivial, I'll trust the result. But for this, you need to have a test set that is large enough for the confidence interval to be significantly smaller than the accuracy that you have in your classification. And if you don't do this, I won't trust the result.
Yeah, I think the effect size thing is a very important point. I also think what Gail was talking about is mostly about inter-subject. We're looking into inter-subject variability, right? And it turns out that in terms of effect size, inter-subject variability, relationship between brain and behavior, these individual differences in brain and behavior, the effect sizes are not that big. But there are a lot of many other applications whereby, for example, within subject analysis, or for example, if you're only looking into what is the brain organization of, what is the brain network structure of different subjects? For that, actually, a relatively small sample size of 20, 30 subjects can probably give you a very good and replicable, let's say, function connectivity matrix, right? So it really depends on the question you're looking at.
Okay, I think 1 minute, one more.
We can do one last one. Yeah.
Maybe the one about other metrics that can be popularized and with the obsession with high accuracy. So so first, accuracy is just one metric. We could have others, and I can even claim that accuracy is a bad metric. There are plenty of reasons why it's a bad metric. First, you can't boil down a scientific reasoning to one metric. That's not possible. But in my view of the world, you do need an observable number that gives you model criticism that's somewhat independent from your modeling choices. And just the fact that you or I have made a complex experiment with a complex acquisition skill and processing doesn't tell me that all my choices were informative. And so I need something independent that tells me whether I'm improving things or not. And accuracy is one choice out of others. It's just that it needs something to capture some form of signal on the left-out population.
But what do you think of, like, in machine learning Twitter as well, there's a lot of complaint about people chasing benchmark accuracy as well. So we could overdo it, I guess, right?
Yes, of course. So I'm on other stages, I've been complaining about chasing benchmarks. It's about being careful not to get in the diminishing returns, right? If you have no improvement in an observable metric, then you need to start asking other questions. And I guess, you know, your figure on the multiple methods, I'm a methods researcher. I've gotten really bored in methods research that shows us improvements that are in the error bars. So I agree. Maybe something that some communities have done really well, and that is happening in this community too, is moving the goalpost. We need to ask more ambitious questions, but once again, then in the different modeling choices that we have, we need to drive them by some observable metric. And it can be something else than accuracy. It just needs to be discussed why it's an interesting metric.
Okay, I think we are already at the end of our session. I guess give us a clap. Unfortunately, I'm also the moderator, so I can say give the speakers a clap.
Should we say high five?